Hello friends, welcome back to a new video that is MCQs on cell biology, and this is the part 2 of the series of videos on cell biology. So the first question of this video, that is question number 21, and the question is a patch clamp device is used to option A measure the strength of an electrochemical gradient, or option B study the properties of individual neurotransmitters, or option C infuse different kinds of ions into an axon, or option D study the properties of individual membrane channels. And the right answer about a patch clamp device is used to option D, that is to study the properties of individual membrane channels, that is known as patch clamp. Okay, so move on to the next question, that is question number 22. That is the distribution of transmembrane protein in the plane of a cell membrane can best be visualized by which of the following? And the options are option A thin section electron microscopy, option B freeze fracture electron microscopy, or option C scanning electron microscopy, or option D SDS gel electrophoresis. So the right one is option B, that is freeze fracture electron microscopy. So the distribution of transmembrane protein in the plane of a cell membrane can best be visualized by which of the following? That is freeze fracture electron microscopy. Okay, so let's move on to question number 23. That is the order of the predicted rate of diffusion across the lipid bilayer, that is highest to lowest, is option A toluene, galactose, phenylalanine, and then chloride ion, that is the option A, or option B, that is chloride ion, then phenylalanine, then galactose, then toluene, or option C, that is galactose, phenylalanine, chloride ion, and then toluene, or option D toluene, phenylalanine, galactose, and chloride ions. So the right answer is option D, that is toluene, then phenylalanine, then galactose, then chloride ions. So the order of the predicted rate of diffusion across the lipid bilayer from highest to lowest is toluene, then phenylalanine, then galactose, then chloride ion. So next question देखेंगे. Next is the correct decreasing order of permeability through a lipid bilayer of the molecules or ions that are isoleucine, tyrosine, O2 and Na plus is option A O2, then Na plus, then isoleucine, then tyrosine. That is the option A. That is the correct decreasing order of the permeability through a lipid bilayer. So the option B, that is O2, then isoleucine, then tyrosine, then Na plus, and option C, that is isoleucine, then tyrosine, then O2, then Na plus, or option D, that is isoleucine, tyrosine, Na plus, and then O2. So the right one is option B, that is O2, then isoleucine, then tyrosine, then Na plus. So the correct decreasing order of permeability through a lipid bilayer of the molecules or ions that are isoleucine, tyrosine, O2 and Na plus is O2, then isoleucine, then tyrosine, then Na plus. It is the decreasing order of permeability. So let's move on to the question number 25, that is all membrane processes such as pumping and channeling of molecules are carried out by option A lipid, option B carbohydrate, option C nucleic acid, or option D protein, and the right one is option D, that is protein. So all membrane processes such as pumping and channeling of molecules are carried out by proteins. Okay, so next question देखेंगे. Next is question number 26. So which of the following statements about membrane transport proteins is incorrect? And the options are option A carrier proteins are similar to enzymes in that they show saturation, or option B carrier proteins can facilitate both active and passive transport, or option C, that is channel proteins can facilitate both active and passive transport, or option D, that is the Na plus slash glucose transport protein carries out secondary active transport. So the right answer is option C, that is channel proteins can facilitate both active and passive transport. So which of the following statement about membrane transport protein is incorrect? That is option C, that is channel protein can facilitate both active and passive transport. So this is not true. Okay. So let's move on to question number 27, that is most abundant lipid in plasma membrane is option A cholesterol, option B sterol, option C glycolipid, and option D phospholipids, and the right one is option D, that is phospholipids. So most abundant lipid in plasma membrane is phospholipids, and that is the option D. Okay, so next question देखेंगे. Next is which one of the following is not a membrane lipid, and the options are option A cholesterol, option B phosphoglyceride, option C cerebroside, option D choline. So the right one is option D, that is choline. So which one of the following is not a membrane lipid? That is choline. So here choline is not a membrane lipid, but other this cholesterol, phosphoglyceride and cerebroside, these are present in membrane lipid. Okay, so next question देखेंगे. Next is the primary thermodynamic factor that favors the formation of a lipid bilayer in aqueous surroundings is option A hydrogen bonds, option B hydrophobic force, option C ionic interactions, or option D covalent bonds. So the right one is option B, that is hydrophobic force. So the primary thermodynamic factor that favors the formation of a lipid bilayer in aqueous surroundings is hydrophobic force. Next question देखेंगे. Next is question number 30. So which of the following lipids is commonly found in biological membranes? And the options are option A monoglycerides, option B diglycerides, option C triglycerides, or option D none of the above. So the right one is option B, that is diglycerides. So which of the following lipids is commonly found in biological membranes? That is diglyceride. Okay, so next question देखेंगे. Next is question number 31. So carbohydrate present on the plasma membrane is option A have structural role, option B form channel, option C act as carrier, or option D help in molecular recognition. So the right answer is option D, that is helps in molecular recognition. So carbohydrates present on the plasma membrane is help in molecular recognition. Okay, next is question number 32. So movement of substances across cell membrane is controlled by the option A size of the permitting particles, option B permeability of membrane, option C membrane proteins, or option D all of the above. So the right one is option D, that is all of the above are right one. So movement of substances across cell membrane is controlled by all of the above. Okay, so next question देखेंगे. Next is question number 33, that is valinomycin, a cyclic peptide, is act as a ionophore, or option Q, that is facilitate the transport of Na plus, or act as a carrier for K plus transport, or facilitates passive transport of cation. So which arrangement is the right one? So option A is P and Q is the right one, or option B Q and S, option C Q, R and S statement is right one, or option D P, Q, R and S all are right statement about valinomycin. So the right one is option D, that is P, Q, R and S. All these options are correct about valinomycin. So valinomycin, a cyclic peptide, act as a ionophore, facilitate the transport of Na plus, act as a carrier for K plus transport, and facilitate passive transport of cation. Okay, so next देखेंगे. Next is question number 34. So in simple diffusion process, molecules cross the plasma membrane. Option A against concentration gradient, option B along concentration gradient, option C do not depend on concentration, or option D with the help of energy. So the right one is option B, that is along concentration gradient. So in simple diffusion process, molecules cross the plasma membrane along concentration gradient. Okay. So next is question number 35. So a passive membrane transport protein, option A, will require a direct source of energy for the transport to occur, or option B, can only transport a molecule down a gradient, or option C, involves a carrier type transport mechanism only, or option D, can move a molecule up a gradient if a membrane potential exists. So the right thing about a passive membrane transport protein is option B, that is can only transport a molecule down a gradient. Okay, so next is question number 36. So diffusion across the plasma membrane is more rapid if a substance is option A protein, option B hydrophilic, option C high in its oil to water ratio partition coefficient, or option D large and globular in shape. So the right one is option C, that is high in its oil is to water partition coefficient. So diffusion across the plasma membrane is more rapid if a substance is high in its oil to water partition coefficient. Okay. So next is question number 37, that is a difference between simple diffusion and facilitated transport is that facilitated transport is concentration dependent, or option B occurs across plasma membrane, or option C requires membrane proteins, or option D utilizes a substance moving with its concentration gradient. So the right thing is about facilitated transport is option C, that is requires membrane protein. So facilitated transport requires membrane protein. So a difference between simple diffusion and facilitated transport is that facilitated transport requires membrane proteins. Okay, so this is the difference from simple diffusion. So let's move on to question number 38, that is erythrocyte glucose transporter specifically transports glucose down its concentration gradient and exhibits hyperbolic saturation kinetics. So this is an example of option A active mediated transport, option B passive mediated transport, option C non-mediated transport, or option D group translocation. So the right one is option B, that is passive mediated transport. So erythrocyte glucose transporters specifically transport glucose down its concentration gradient and exhibits hyperbolic saturation kinetics. This is an example of passive mediated transport. Okay, so next question next is question number 39, that is primary active transport. Statement 1, that is P, requires carrier proteins. Option Q, that is transport solutes against concentration gradient or coupled to ATP hydrolysis, or option S, that is driven by movement of a co-transported ion down its gradient. So which statement about the primary active transport is correct? So options are option A P Q R, option B P and S, option C Q and R, or option D P Q R and S. All statements are correct about primary active transport. So the right one is option A, that is P, Q and R. These three statements are correct. So P means requires, this primary active transport requires carrier proteins. Then this primary active transport transports solute against concentration gradient and coupled to ATP hydrolysis. These three are correct about primary active transport. Okay, so the last question of this video, it is question number 40. So fluorescence recovery after photo bleaching, that is FRAP technique, is typically used to measure the rotational motion of membrane lipids and proteins, then or is typically used to measure lateral diffusion of membrane lipids and proteins, or option R, that is involves fluorescent labeling of cell surface molecules, or statement S, that is has demonstrated that lipid diffuse freely over short distances but generally not over longer distances in fibroblast membrane. So the right one is option. So the options are option A P and Q, option B P Q R, or option C Q R S, or option D R and S. So which statement is correct one? So the option C is the correct one. So that is Q, R and S are correct one. So Q, R and S, these three statements are correct about fluorescence recovery after photobleaching, that is FRAP technique. So this FRAP technique is typically used to measure lateral diffusion of membrane lipid and proteins. Then it involves fluorescent leveling of cell surface molecules, and it has demonstrated that lipid diffuses freely over short distances but generally not over longer distances in fibroblast membranes. Okay, so these are all 20 questions of this video on cell biology. Okay, so thank you for watching this video.